And there are various steps in transcription. First of all, you have to understand. Can you tell me what is coding strand? What is
It's
coding strand?
the strand that codes for the DNA. I mean,
And what is template strand?
the one that the RNA copies from.
Yes. Okay. So whatever the code is written on the opposite strand of template strand is almost similar you will see on the mRNA. Okay.
Mm-hmm.
So here is the example that this is the DNA. Okay. So this one is the DNA strand. This is the DNA strand. This is the DNA strand. If this one is the template strand, and always remember that 3 prime to 5 prime end is always work as template strand. Okay.
Mm-hmm.
Always remember this 3 prime to 5 prime end is work as a template strand. Okay. So this template strand is the strand which provides the information of nucleotide sequence about the on the mRNA. Okay.
Mm-hmm.
So if you see that the T complements with A complements with U, C complements with G, T complement with A complement with U, and G complement with C. And if you see that this A is here, G is here, A is here, and C is here. But as this is a DNA strand, this uracil is just replaced with T, and this uracil is replaced with T. So we can say that all this nucleotide sequence is almost identical to the sequence which is present on the opposite strand of DNA. That's
Mm-hmm.
why this is called the coding strand. Coding strand means the code which is written on the mRNA. Okay,
Yeah.
understood?
Yeah.
Okay, so there are various steps in the transcription. First one is initiation. Okay, first one is initiation.
Mm-hmm.
Okay, first step is initiation. In this step, RNA polymerase enzyme binds to the promoter region on the DNA. Do you know what is promoter?
Um. No.
Promoter region on the this is the coding strand and this is the template strand. Okay,
Mm-hmm.
so this is the region where you can say that promoter is the region where from where transcription starts. Okay, if someone asks what is promoter, the promoter region, a promoter is a region on the DNA, that stretch of DNA that initiates the transcription on gene. Okay, so this is the region from where transcription starts. Okay, that's why RNA polymerase here at this position, RNA polymerase will bind at this region, at promoter region. Okay, and
Mm-hmm.
this is the terminator region. Always you will see that promoter region is present at the at which end 5 prime end of you, or you can say that 3 prime end of template strand. You can say that promoter is present at either 3 prime end of template strand, or you can say that at 5 prime end of coding strand. Okay, both answers are correct. Understood? So
Yeah.
first of all, you have to understand initiation step. Initiation is the step during which RNA polymerase binds to the promoter region on DNA. Okay, so in the initiation step, first of all, RNA polymerase binds to promoter. Okay, if someone asks where you will find the promoter region, you can tell that It is found at 3' end of template strand, or you can say that it is found at 5' end of coding strand. Okay.
Mm-hmm.
Now, the promoter signals the start of transcription. Okay. Promoter signals the start of transcription. After then, transcription will start. Any doubt?
No.
If you do not understand about the promoter, if you do not know about the promoter, then you will not understand about the how gene will produce the enzyme. Okay.
Mm-hmm.
So you need to know about the promoter. So the next step is elongation. Okay. The next step is elongation. Elongation, RNA polymerase synthesize RNA by adding complementary ribonucleotides to the growing RNA chain. Okay. So as the RNA polymerase moves at 3' to 5' end on the template strand, RNA starts get polymerized and different types of nucleotides get coded on the mRNA. And those nucleotides are adenine, uracil, guanine, and cytosine. Okay.
Mm-hmm.
RNA during elongation process, RNA polymerase synthesizes RNA by adding complementary ribonucleotide to the growing RNA chain. Complementary to which strand of DNA? Can you tell me?
Template strand.
Yes, template strand. Okay. Now, during elongation process, you will also see that the strand being synthesized is called the primary transcript. Okay. So this strand is called primary transcript. This strand is called primary transcript. Okay. During the elongation process, the mRNA which gets synthesized is firstly known as primary transcript. Okay.
Mm-hmm.
Then there is a third step is called termination. Okay. Transcription ends when RNA polymerase reaches a termination sequence. This is the termination sequence. Okay. So every gene has unique promoter sequence and unique terminator sequence. Okay.
Mm-hmm.
During your further studies, you will see that you will study about every gene has different types of promoter and different types of terminator. Okay. I think you didn't studied about that promoter right now.
Not yet.
So the third step is termination. Okay, the third step is termination. And during this step, transcription ends when RNA polymerase reaches to which sequence? Terminator sequence. Okay,
Mm-hmm.
and this terminator sequence is also unique sequence. Okay, and the RNA transcript detached from the DNA template. So this RNA transcript, or we can say that this RNA primary transcript get detached at the terminator end of transcription. Okay,
Mm-hmm.
any problem?
Mm-hmm.
So what you will see that only one strand of DNA is transcribed. So RNA have which transcribed sequence of the DNA, the template strand. Okay.
Mm-hmm.
And the second main point during this transcription process is the coding strand has the same sequence as the RNA, except T is replaced by U. Okay.
Mm-hmm.
So the first important point is only one strand of DNA is transcribed. That is template strand. Okay. Second important point is the coding strand has the same sequence as the RNA, except T is replaced by U. What is T?
Uracil.
No, what is T? Which
Oh,
nucleotide is
uh,
symbolized as T?
thymine.
Yes, thymine. Thymine is replaced with uracil. And the third important point is that you should know that in prokaryotes, transcription and translation occurs simultaneously. Okay,
Mm-hmm.
transcription and translation occur simultaneously. Can you tell me why?
Um, I don't know.
Because prokaryotes do not have well-defined nucleus.
Oh,
Okay,
hmm.
and
So
their
they occur
nucleus, yes, tell me.
they occur simultaneously in prokaryotes or eukaryotes?
Yes, no, in
Prokaryotes.
prokaryotes,
Okay.
because prokaryotes do not have well-defined nucleus.
Mm-hmm.
Okay, so transcription and translation occurs simultaneously in the prokaryotes, but in eukaryotes, first transcription will occur inside the nucleus of cell. Then, after few modification on the mRNA, okay, after few modification on mRNA, you will see that the translation process will undergo inside the cytoplasm of the cell. Okay, so
Mm-hmm.
if I will quote that, what is the main difference of transcription in prokaryotes and eukaryotes? First thing is RNA polymerase. So in prokaryotes, single RNA polymerase will work. In the prokaryotes, only one type of RNA polymerase is working. And in the case of eukaryotes, three types of RNA going to work, that is RNA polymerase 1, 2, and 3.
Mm-hmm.
And if I guess you do not studied about that.
No.
Okay. And also, just keep it in your notes.
Mm-hmm.
Uh, During our sessions, I will teach you in detail about these things.
Mm-hmm.
Okay. So, RNA polymerase in prokaryotes, single RNA polymerase will do transcription. But in the case of eukaryotes, three types of RNA: polymerase 1, polymerase 2, and RNA polymerase 3. Okay.
Mm-hmm.
In prokaryotes, transcription is very simple. Okay. But in eukaryotes, there is a very complex process of transcription. And I have already told you that in prokaryotes, there is no any post-transcriptional modification. Why?
Mm-hmm.
Because transcription and translation is going simultaneously in prokaryotes. But in eukaryotes, you will see the post-transcriptional modification. That is capping, polyadenylation, and splicing processes.
Mm-hmm.
Okay.
Yeah.
So, do you ever heard about these terms, capping, polyadenylation, and splicing?
No.
Okay. So, what I have uh, lectured you right now, is it related to your provided notes in your classroom, or I have given more than that or less,
Um, the things that
or
we've
I have missed
done,
anything?
I don't think we've really gone over like um the specific like differences between prokaryote and eukaryotic transcription. I think we've been mainly
Okay.
focused
So,
on eukaryote.
okay, in eukaryotes.
Mm-hmm.
So, what are more things present in your notes? Did anything I have missed?
Uh, on transcription, I
Yes,
don't,
on transcription.
I don't think so. We learned in my class, we learned that the proteins determine most characters of a cell and organism. The information stored in the DNA determines what protein can be made on by a cell. The environment influences which proteins are made by a cell. Um, Ribosomes make proteins but are not in the same location as DNA in the cell. And yeah, like
More, is there more in it? uh, in your notes?
Um, for transcription, I think that's it.
Well, let's continue on translation, then I will give you.